Start করুন।
Oh, okay. Good evening to all of you. I hope that you have done a very good exam. The questions were easy, and I will be discussing today biology section of the code M6, and from the question number 136 to 150. So let's start with it. Question number 136, section B. In the exponential growth equation, NT equals to NOERT, where E represents. Yes, you know that it is very important and very frequently asked, and that means the equation is very known to all of you. And this is basically the base of E represents the base of natural logarithm. This is also there in the NCERT book. I would like to share the page. It is a direct question from the NCERT, and here it comes with, that is, I hope that you can see this page. Just a minute. Yes, this is the equation of page number 230. It is a direct question where you know that NT is a population density after time t, NO is the population density at time 0. R is the intrinsic rate of natural increase in this equation. And here E is the, yes, E is the base of natural logarithm. So the correct answer for this question will be base of natural logarithm. That is question number 136. The answer will be 3. Now let us move on to another question, the next question rather. Okay. After 136, we move on to question number 137. Yes, it is a match the column. Match the following or match the column. Here you can see nitrococcus. In column 1, it is given nitrococcus. And that is nitrococcus. It is going to match with, that means conversion of ammonia to nitrate. We know that amino acid to ammonia, it is ammonification. Then ammonia and all these things, they get converted into nitrite, and ultimately then they get converted into nitrate. Here we see that the nitrococcus bacteria is responsible for, we know that it is responsible for conversion of ammonia to nitrite. So A will go with 2. Then next we see that rhizobium is there. Yes, rhizobium is responsible for conversion of atmospheric nitrogen into ammonia. So B is going with 4. And thiobacillus, it is a very popular denitrifying bacteria. So C is going with 1. That is denitrification. And last option, that is nitrobacter. Yes, nitrobacter, it is responsible for conversion of nitrite to nitrate. So it will go with 3. So the option 1 for question number 137 is the correct answer. Clear? Next, move on to question number 138. Just a minute. Now, in the question number 138, we can see that it has been asked from cell cycle, the different events which happens in the cell cycle. In the list 1, we have to match list 1 with list 2. Here in the list 1, the option A is S phase. And we know that, yes, we know that S phase, it is responsible for DNA replication because nucleic acid replication occurs in S phase. So A will go with 4. Then G2 phase, it is concerned with synthesis of different substances, especially proteins. And these proteins are required during the M phase or divisional phase. So G2 will go with 1, that is proteins are synthesized in the G2 phase. Yes. So we can say that G2 will go with 1. Then quiescent stage. We all know that this quiescent stage is a stage. It is called perpetuating interphase. Or you can say that it is a phase where the cell gets suspended. They goes out of the cell cycle. They remain suspended. And also they become inactive. So here, quiescent stage will go with option 2, that is the point 2 of the list 2, that is inactive phase. And Then we see that G1 phase, it goes with option 3. That is, it is an interval between mitosis and initiation of DNA replication, because G1 is the phase of interphase which is present between mitosis and S phase. So the correct option for this question, that is question number 138, is option 3. Now let us move on to question number 139. Now here in question number 139, it is also a direct question from NCERT. Here, you have to identify the correct statement. And this question is related to the RNA modification after transcription. The first option is capping methyl guanosine triphosphate is added to the 3' end of the HNRNA. We know that capping occurs in the 5' end, that is methyl guanosine cap. Yes, 7-methyl guanosine cap is attached to the 5' end of the HNRNA, so that it is protected from the 5' exonuclease activity. So this statement is not correct. Next, we move on to the second statement. RNA polymerase binds with the Rho factor to terminate the process of transcription in bacteria. Yes, this is also correct, because during termination of transcription, we know RNA, basically we know there is a Rho dependent or Rho independent termination is possible. In case of Rho dependent termination, there is a factor called Rho factor, which behaves as a helicase. That means it has an ATPase activity which binds to the RAT site of the RNA, hanging RNA, and goes and then gradually it moves and binds with the RNA polymerase. As soon as it binds with the RNA polymerase, the termination of transcription takes place. So this is also a correct answer. This is also a correct statement. Third statement, coding strand in transcription unit is copied to an mRNA. This is also correct, because we know during transcription, the DNA have two strands, obviously. One is known as the template strand or non-coding strand, while the coding strand is the one which have their sequence similar to that which is present in mRNA. Only the thymine is replaced by uracil in mRNA synthesized, because coding strand and non-coding strands are complementary to one another. And on this template strand or non-coding strand, RNA synthesis takes place. So this RNA, which is synthesized on the basis of complementarity on the 3' to 5' strand, that is the template strand, you can also call it as the, that is non-coding strand, will have the same sequence which is present in the coding strand. So this is also correct. So let's come to the fourth statement, that split gene arrangement is a characteristic of prokaryote. So split gene, we know that prokaryote do not have any exon and intron. Yes, they do not have exon intron. In case of eukaryote, the gene is splitted and it contains exon intron in this manner. So that is why split gene concept is stipulated to eukaryotes and it is absent in prokaryote. So this option 4 is the wrong statement, and in this question identify the correct identify the correct statement is given. So in question number 139, the correct answer, identify the correct statement, it will be option 4. Next, we are moving to question number 140. That is the plasmid PBR 322 has PS1 restriction enzyme site within the gene ampicillin resistant that confers the ampicillin resistance. If this enzyme is used for inserting a gene for beta galactosidase production and recombinant plasmid is inserted in E. coli strain. So what will be the answer for this? That means when we see that a particular gene is introduced over here, say this is the diagram, you can see that ampicillin, it is cut by PS1, and when a foreign gene is introduced for beta galactosidase, the bacteria cannot synthesize or would not be, that means it won't be able to resist against the ampicillin resistant genes. Ampicillin resistant genes. So the correct answer for this question is question number 140. Is 1. It will not be able to confer ampicillin resistance to the host. Next question, DNA fingerprinting involves identifying differences in some specific regions of DNA sequence called, yes, it is a repetitive DNA. It is also a direct question from NCERT. Question. Just a minute. Then next we are moving on to the another question, that is question number 144. That is, nowadays it is possible to detect. Okay. Nowadays it is possible to detect mutated gene causing cancer by allowing radioactive probe to hybridize its complementary DNA in a clone of cells, followed by detection using autoradiography. Now, why it happens? That means autoradiography, because mutated gene partially appears on the photographic film, mutated gene completely and clearly appears on a photographic film. Mutated gene does not appear on the photographic film as the probe has no complementarity with it. So the answer for this question will be option 3, because mutated gene does not appears on the photographic film as the probe has no complementarity with it. Next question, which of the following statement is incorrect? Yes, both ATP and NADPH are synthesized during non-cyclic photophosphorylation. That is correct. Stroma lamellae have PS1 only and lacks NADP reductase. That is also correct. The grana lamellae have both PS1 and PS2. Yes. The cyclic photophosphorylation involves PS1 and PS2. No, only PS1 is involved. So for question number 145, it is option 4. Then question number 146, which of the following statement is incorrect? During aerobic respiration, the role of oxygen is limited to terminal state. That is correct. In electron transport chain, one molecule of NADH give rise to 2 ATP molecules, no, 3 ATP molecules, and 1 FADH2 gives 2. So this one is incorrect. So you have to find out the incorrect statement. And this is the incorrect one. That is for question number 146. The correct answer is option 2. Now question number 147. You again have to match. Protein is matching with, yes, peptide bond, unsaturated fatty acid CC double bond, nucleic acid phosphodiester bond, and polysaccharide with glycosidic bond. So for the correct answer is 147 is one option, first option. Which is the role of RNA polymerase 3 in the process of transcription in eukaryotes? RNA polymerase 3 is responsible for synthesis of tRNA, 5S rRNA, and snRNA. So the option 2 is correct. And then we can move on to question number 149. In 149, again, we have to match the column. That is, large colorless empty cells in the epidermis of the grass leaves are subsidiary. No, subsidiary cells are cells which are, that means, which are basically associated with the stomata. That means those cells which are associated, they are called subsidiary cells. In dicot leaves, vascular bundles are surrounded by large, thick-walled cells. They are not called conjunctive tissue. They are called bundle sheath cells. And cells of the medullary rays that forms a part of the cambial ring is interfascicular cambium. This is correct. Loose parenchymal cell rupturing the epidermis and forming a lens-shaped opening in it is called spongy. No, it is also wrong. So the correct option is option 3. The last question, that is, some members of which of the following pairs or families of pollen grains retain their viability for months? Yes, it will be applicable for Rosaceae, Leguminaceae, and Solanaceae. For this, option number 4 is the correct answer. So I hope that you have done all these things correctly. So thank you.